Have you ever pondered about the possibility of modifying the genetic code of an organism? The ability to add, remove, or alter specific sections of the DNA? Welcome to the intriguing world of gene editing, a realm where science fiction meets reality. This revolutionary process allows us to make precise changes to the DNA of an organism, akin to editing a script. But the script in this case is life itself. Gene editing is a tool that provides us with the capability to manipulate the genetic code, the fundamental building blocks of life. It enables us to add, remove, or alter specific sections of DNA, thus paving the way for unprecedented advancements in the field of genetics. One such groundbreaking technology that stands at the forefront of this revolution is CRISPR-Cas9. This innovative tool has turned the tables, enabling scientists to modify genes with an accuracy and efficiency that was once deemed impossible. Indeed, the realm of gene editing is like a vast ocean of possibilities waiting to be explored. So you may wonder, how does this fascinating process of gene editing work? Well, allow me to shed some light on this captivating subject. Imagine a long chain of building blocks, each unique and vital to the construction of a magnificent structure. This is much like our DNA, a string of genes, each contributing to the incredible architecture that is life. Our genes determine everything from our eye color to our likelihood of contracting certain diseases. Now, what if we could swap out some of these blocks, adding, removing, or altering them to create a different outcome? This is where gene editing comes in. At the heart of this process are tools like CRISPR-Cas9, a revolutionary technology that has turned the world of genetics on its head. Think of CRISPR as a pair of molecular scissors. Scientists program these scissors to locate a specific sequence in the DNA. Once there, CRISPR makes a precise cut, effectively breaking the DNA strand. Now here's where it gets truly interesting. Once the DNA is cut, the cell's natural repair machinery kicks in. This is a process that the cell routinely does to fix any breaks in the DNA. However, while the cell is busy mending the break, scientists can introduce changes to the genetic code. They can add a gene, remove one, or even alter an existing one. Let's put this into context. Say there's a harmful mutation in a gene that could potentially cause a disease. By using CRISPR, scientists can locate this faulty gene, snip it out, and replace it with a healthy version. Just like that, the risk of the disease is significantly reduced. This is the beauty of gene editing. It's precise, efficient, and holds immense potential. It's a game changer in fields ranging from medical research to agriculture and biotechnology. In essence, gene editing is akin to editing a manuscript, but here the manuscript is the blueprint of life itself. Now that we understand how gene editing works, what could we use it for? Well, the applications of gene editing are wide-ranging and revolutionary. The most significant advances are occurring within the realm of medical research, particularly in the treatment of genetic disorders. Imagine a world where we can rectify the genetic anomalies that cause diseases like cystic fibrosis, sickle cell anemia, or Huntington's disease. By precisely targeting and modifying the problematic genes, we can potentially eradicate these conditions. This isn't a far-off dream. It's a reality that scientists are striving to achieve every day, but the impact of gene editing extends far beyond the healthcare industry. In agriculture, gene editing can help us create crops with desirable traits. We're not just talking about bigger, juicier tomatoes here. We're talking about crops that are more resilient to pests, diseases, and extreme weather conditions. By tweaking the genetic makeup of these plants, we can enhance their nutritional value, extend their shelf life, and increase their yield. In essence, gene editing can play a pivotal role in addressing food security issues and promoting sustainable farming practices. And then there's the field of biotechnology, a realm where gene editing is opening up a world of possibilities. It's paving the way for the development of new products, from biofuels to biodegradable plastics. Think about bacteria engineered to eat up oil spills, or yeast that can produce bioethanol. These are not just pipe dreams, but tangible solutions that can help us tackle some of our most pressing environmental challenges. Gene editing is not a tool to be wielded lightly, but it is one that holds immense potential. It's a technology that can help us rewrite the blueprints of life to create a healthier, more sustainable future. It's a testament to human ingenuity and our ceaseless quest for knowledge. From medicine to agriculture, gene editing holds the potential to transform numerous fields. And as we continue to unravel the intricacies of our genetic code, who knows what other doors we might open? We've delved into the world of gene editing, but what are the key takeaways from our exploration? In our journey, we've unraveled the intricate process of gene editing. It's a fascinating field that involves making precise changes to the DNA of an organism. This can entail adding, removing, or altering specific sections of the genetic code. The advent of technologies like CRISPR-Cas9 has revolutionized this sphere, allowing scientists to modify genes with an accuracy and efficiency that was once the stuff of science fiction. We also walked through the mechanics of gene editing. It's a complex process, but at its core, it's about using a molecular scissors to cut the DNA at a specific location. This cut then triggers the cell's natural repair mechanisms, which can be harnessed to introduce the desired changes to the genetic code. This precise targeting and manipulation of genetic material is what sets gene editing apart and makes it such a promising tool. Beyond just understanding the process, we've looked into the vast applications of gene editing. It's not just a technology for the future, its impact is being felt here and now. In medical research, it's opening up new avenues for treating genetic disorders and fighting diseases. In agriculture, it's helping us create more resilient and productive crops, while in biotechnology, it's driving innovations that are transforming industries. Yet, as we've discussed, gene editing isn't without its challenges and ethical considerations. The potential to rewrite the genetic code of organisms, including humans, is a powerful tool. But with this power comes a profound responsibility. How we wield this tool, how we navigate the ethical dilemmas, and how we balance the potential benefits with the possible risks will shape not just the future of gene editing, but the future of humanity itself. As we stand on the brink of a new era, gene editing presents us with a powerful tool, but also a profound responsibility.